We're here. We're ready. We're starting. Nice, very animated discussions this morning. Nothing like a little bit of snow to make everybody so happy. Ugh. Okie dokie. Where I left off. Aha. So hello out there. Okay, I sent you this. I sent you the study guide this morning. Okay, and I have no idea when this exam is going to be. Let me get it. I have to finish this chapter and start the next chapter to have sort of a read on that kind of information. That so don't get overly excited yet about that. So let us begin. We kind of we're at ribosomes when we kind of left off, and so it's a good place to start. And this is going to be a very interesting because of the distinction between the bacterial and eukaryotic ribosome, as we'll see brought forward into the next chapter. Again, it's a significant target point for the use of antibiosis, and certainly, I mean, again, we have a living thing. It does what we do. It's just a different structure of it's a little more simplistic, I would dare say, really more so than anything else. Because we don't have the elaborate membrane-bound organelle structure in bacteria. Basically, they make proteins, and the proteins, but most of the time, the proteins are doing not a whole lot more than setting up the stage for reproduction, and maybe when they're growing, secreting agents that, for you and I, are toxic if they're pathogenic bacteria. And if they're useful bacteria, then it's just a useful situation that those particular agents are harmful to other bacteria. So as you're going to see, they're more or less, I hate to say it this way, the bacteria are much more of our friends. They get a bad rap. Okay, viruses are not. But bacteria, the vast majority of them occupy spaces on us to prevent other agents from attaching. They break down the oils and things that you remember learning about in skin, and basically they create what we call an acid mantle. Okay, that's there. It really restricts other types of agents, particularly other types of bacteria, from gaining a foothold and creating problems for us. So by and large, I mean, I think that's the misconception that we have about microorganisms. We're covered in them. You did the last week that lab where you do the environmental sample. Did you do skin samples and stuff like that? Okay. You didn't do skins. What are we doing? I always had people, behind their ears and under their arms and all those different kinds of places to look. We're covered. We are completely covered in microorganisms. If something came down from outer space that could see us, they'd look and they say, "Oh my God, there's this organic thing and it's covered with all these other organic things. This is what we look like." Okay? Which might be an improvement I think for some, but I digress. Nevertheless, that's really where it is. So we'll start here. So for our purposes, 7DS is the ribosome. Don't care about the subunits. 80S for eukarya. More on that down the road. They merge together. The S, again, stands for a scientist named Svedberg, capital Svedberg, and it's just a matter of, it's a density unit. It's not angstroms and things like that. Scientists love to name things after themselves. Most of these kinds of things are useful inclusion bodies and microcompartments. We use them more in the laboratory for identification, and they do give us some hint about what things are. So what you're going to see is that certain organisms have a greater affinity for oxygen. Perhaps they're photosynthetic. Even in bacteria, we have unusual photosynthetic bacteria. They have gas vesicles, so they're able to float in the system. You're going to see tiny little magnetite particles, okay? And there's some that are attracted or repelled by certain kinds of magnetic current. I mean, so there's really unusual ones, and we really are able to utilize them more for identification purposes. So again, the iron oxide with magnetic, the gas vesicles that are there, all of those types of things. So they're arranged in some sort of geometrical fashion. I don't think it's really a big deal for us to go into the fine points that are there. We used to think there was no cytoskeleton, as there is. It was such an elaborate cytoskeleton that we all studied in AP1. But some bacteria produce these long polymers similar, okay? Ribbons play a role with regard to shape. You see them in all forms. Uh, down to it. And again, why do we study these things? We always look at, and you're going to see, why do we spend so much time looking at the replication cycle of viruses, looking, as we will in subsequent chapters, at the life cycle of bacteria? Because that's where you attack things. You look for how they do business and you attack them. I mean, it's in any strategy that's there. If you're like me and just watched every conceivable football game in over the last couple of days, what are they looking for? Where the defense can be attacked, where the offense can attack the defense. That's all it is a strategy type of thing. And that's what in science we use a lot of those things. We look at things that harm us and try to analyze how we can take it. It's like taking snake venom and refining it and using it isolatedly to attack certain cells that might be problematic. I mean, that's what you do. You find something that's unique or unusual and you try to slave it, as it were, to your abilities, or to something that will be beneficial to us. Okay. The endospore. There is an odd bacteria called Sporosarsina that we really don't talk about so much, mostly because I don't know that it's necessarily as well understood. But for our purposes, these are the two genera of bacteria that form endospores that are relevant to us. Bacillus, with a capital B, again, and Clostridium, which I talked about in the very first week with regard to tetanus and botulism. The distinction between them is very simple. Bacillus is aerobic, so we can study the endospores in the laboratory. Endospore forming bacteria in the Bacillus family are useful because they produce some types of strains of antibiotics. Most notably, the one you work with in the laboratory, Bacillus subtilis, which you're gonna cultivate, and you're gonna probably endospore stain. That's there, and we're fond of calling it B sub for short. It makes something called bacitracin. Bacitracin is one of the three components of the original formulation of triple antibiotic ointment. Neosporin, which we're going to study in antibiotics, polymyxin B, also we're going to study, and bacitracin. Okay, they're all so strong we can't use them internally, so they're used topically. Where was that formulation developed? That's right here in Pittsburgh, Burroughs Wellcome Company. Just a little Pittsburgh, from a New Jersey guy, a little Pittsburgh trivia. Okay. So Clostridium, more because it's anaerobic and it causes those notable conditions we discussed in the very first week, in botulism and tetanus and gas gangrene and C. diff, but it has to form in areas that are bereft. They don't have any oxygen. Okay. So first, the most confusing thing for students about endospore formation. Bacteria are vegetative forms. If you're in a permanent, and heaven forbid you are, a permanent vegetative state, you're sitting there on machines doing nothing, right? Okay. She's in a permanent, or he's in a permanent vegetative state. Okay. For bacteria, it's the complete opposite. Remember, we thought they were plants. When bacteria are vegetating, they're having a good time. They're reproducing. They're living. They're doing what they're supposed to do. They're making more bacteria. So a vegetative bacteria is a normal, flourishing, reproductive bacteria. Whether it's helpful or harmful, don't matter. The endospore stage that can be accomplished by these limited groups is a state, for lack of a better way to express it, of suspended animation. Okay, it's going into a form that is nearly indestructible. Okay, so they've done studies where they've traced them back untold millennia, and they're constantly present. That's the thing I told you about with things like tetanus. It's on the bottom of your shoes. It's in the soil. That's where you find them most frequently. So it's always there. And indeed, when we take the next step and look at methods of control and sterilization, the whole idea is to destroy these endospores. So sporulation is induced by environmental conditions. For most bacteria, that means an absence of nutrients, typically carbon and nitrogen-based nutrients. They don't have enough. That's there. That's the major adverse condition. Lack of water would also be in there because water is essential for all living. So you have those two things that are part of it. So if you starve the bacteria, okay, and that's why we let them, they typically grow better on plates in the laboratory because eventually the nutrients are gobbled up. Is really the best way. They grow a little less ably in tubes of broth that you sometimes use in the laboratory. And you'll even notice that with B sub, the cultures typically form little crusts on the upper part. They're very aerobic, so on the upper portion of the liquid, and they kind of form like almost a solidified layer. I forget the microbiological laboratory name of it, but it'll come to me sometime in the future. So nevertheless, this is what this is about. Endospores resist extremes of heat, drying, freezing, radiation, chemicals that would kill other cells. Okay, they show you this. It's hard. We'll do the animation. It's much better. I've been using this animation, I don't know, for 25 years. So I'm trying to crank up. That's going to be too loud. And you'll find this on that zip drive that I had before that has all these animations. That's on Blackboard. And I think I even loaded these MP4s specifically. So let's see if this, I mean, we look good in play.
As vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolinic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
So effectively what happens in this, without going through it in chapter verse, I mean, if it's replicating, it's just dividing in two like that. But when the conditions are adverse, whatever those are, and typically it's nutritional, what happens is we see this irregular division, not in terms of DNA. Why is this not a form of reproduction? Because it's binary fission is reproduction in bacteria. You have one, now you have two. Here you have one, and at the end, you still have one. Okay, because what happens is one of the two copies, as you can see there, deteriorates. So as effectively the bulk of the mother cell envelops that forespore and the mother cell's DNA degrades, we just have one set of DNA, just like we started. Okay, now within that double membrane, and all of these to our knowledge are gram-positive, a relatively thick layer, read that as protective, forms. And then proteins are attracted. So now you have this protein coat. Interior, interestingly, what happens is this compound called dipicolinic acid does two things. It pulls in calcium to harden it, and it extrudes water, effectively, if you will, to dehydrate it. Okay, so what it's doing now is you just have a sufficient amount of cytoplasm to envelop the DNA, and really this very, very hard shell with additional proteins and sometimes an additional coat forms. And typically when you see them in the lab, some will be within the cell and some will be already opened up or lysed and be in the environment. What's interesting is the positioning of the spore gives you a clue as to its identity. So in B sub, the spores are typically in the middle of it. Where you're going to see, like in Clostridia, typically the spores are more to the end. They look kind of like a tennis racket shape. And so that plays a role in the lab identification. It's not lab. I'm not going to ask you about that, but because I don't think you're going to do. We typically don't do anaerobic bacteria here. They're a little hard to cultivate. You end up having to use a chamber and you activate a few elements to sort of suck out the oxygen. And if you do it just right, you can grow that stuff in there. We have the materials to do it. I just don't think we do it that frequently. I mean, I don't typically do it when I do the lab. So that's the endospore nugget. And I mean, if you want to go through these slides, go ahead. I mean, it's really telling you the same thing about it's replicated, the envelopment. They don't focus too much on the deterioration. And here's kind of what's left. That's there. Anthrax, tetanus, gas gangrene, botulism, C. diff. I mean, most notable ones. There's a lot of other bacilli. What they do, as you will see in the hospital, is they take a tame bacillus. That's the hardest one to destroy. And it's got a fancy name called Bacillus stereothermophilus. Don't you know that? And they put that in little packets when they sterilize, and they put that in because it's a way of calibrating and making sure, verifying that your sterilizer is working. That's what they do in the hospital. And then what they do is they incubate them. If they grow something, it's bad. It's a bad run. In hospitals, it's very much about doing it properly, because the fact that something says it's sterile, you have to do additional tests that are there. I'll get into that when we talk about sterilization. We used to do all our own sterilization in the office because I did probably 90% of my procedures in the office. And you treat it, you just dedicate a room and treat it like an operating room. And many of you, if you're going into nursing, these days, a lot of outpatient stuff. Infection rates are lower outpatient, inpatient. One thing about hospitals that they all have in common: sick people. Just go. I figured that out on my own. I'm sorry for being so talkative. I spent the whole weekend with talking to the dog and 2 cats. Just my wife staying with her mom. Where in the bacteria did you find the genetic material? In the nucleoid, of course. Compare and contrast, we do a lot of that. The archaea, I don't spend a lot of time in. With regard, some of the ribosomal RNA was what it was used initially when the domain theory was put together. Okay, so there's a lot of similarities that are there. They have very unique RNA, different the way the DNA compacts itself. And to me, that's not important for us. What is important, and the membrane structure, which doesn't have peptidoglycan, but the fact that there are extremes is very, very significant. We do know that there are some that conceivably cause disease, but they're not included for the sake of completion in this particular course. 70S ribosomes. We're not doing the whole thing with the Berge's manual and the different types of firmicutes and things like that. Species just are obviously a member of the generic, gives you the general characteristics. The species are the individual. So you're going to see when in the lab, you're going to work with Staphylococcus aureus, which is a pathogen, and Staphylococcus epidermidis, which is on all of our skin all the time. And it can be a pathogen if somebody is debilitated or compromised, those comorbidities things. Berge's manual, whether they comment on it in the lab or not, is when you're trying to do, and what your lab is, a bacteriology lab, because we really don't study viruses, and they can grow some fungi. But by and large, most of your lab works on bacteria. So I would call it a bacteria lab. And Berge's manual is sort of like, for lack of a better way to express it, a biological barcode. Okay, so when you try to identify bacteria in the lab, is it gram-positive or gram-negative? Is it an endospore form or not? Okay, those kinds of things. Does it have a capsule or not? Does it have flagella or not? Does it ferment certain sugars? Does it not ferment certain sugars? Is it aerobic or anaerobic? And it's like yes or no, yes or no, yes or no. And Berge's manual of determinative bacteriology does just that. And you can look at that, and they'll even subspeciate it. So one species will do that, and one will not do that. And that's kind of what you do in the lab. And I don't know if Dr. Dress and Dr. Hilwig are doing this, but typically I do unknowns in the lab. I don't know if that's part of your syllabus or not for the lab. To me, that's kind of fun. You kind of do them as a team, and it's fun. And here it is. Yeah, views. Yeah, yeah. And these are the cutes, and we don't do those. Uh, like, wouldn't they be bacterial shaming if you had a thin cell wall versus a thick cell wall? Right? Can you see somebody on social media doing that? You have a very thin cell wall. I'm on a diet. Isn't that clever? Thanks. Okay. Shut up, Lou. Nobody cares. Overall similar pattern. 90% okay. Some species are even characteristics within that. Serotyping is something that's extremely important, just to mention in passing. I would think you'll get more of that in clinical things. One of the ways we identify things is we look at the antibody. Serotyping means we're checking to see for the presence of either a particular antigen or an antibody. When someone's HIV positive, okay, what has caused that, or Hep C positive, is they have produced antibodies to that virus. So we know that at one point in time has invaded them. Whether it still has a presence, whether it is still active, is another question entirely. Then you do blood tests or other fluid tests to look for the antigens and do effectively RNA or DNA identification, typically PCR, which is a means that was developed based on microbiology in any event. Okay, so that's part of the stuff. I think it's important, at least for context, that unique surface molecules, every living thing and every chemical for that matter, basically has very unique characteristics that we respond to. If you did not know, when it comes to biomaterials, just to give you an idea, the reason that they implant for years, now there's a lot more advances in material science when when they put wires or screws or pins, initially they're made out of stainless steel, very low reactive. They use a lot of polymers, a lot of plastics, very low reactivity. There's not a lot of surface molecule. That's why you use certain things. For years, when we sewed people, we used silk. Silk is very reactive. So then they started to use a lot of synthetic polymers for suture material or materials that would dissolve. For years, when I first started, we used what was called catgut. It was literally catgut. It was treated and sometimes coated. So you had plain catgut and chromic catgut and silk. That's right. 45 years ago, that was the standard. Not good for the cat. Yeah, I don't care. Good, done. So much for that. I was getting tired of it. It was getting boring anyway. This presentation was going nowhere. Okay, so let me back up a little bit. This is the Nestor PowerPoint, which I included. And I just want, and it has basically the same things. Again, these are the different, again, if it's gas vesicles, those little magnetite particles, some are attracted to heat, some are attracted to light if they're photosynthetic, for instance. Again, the pili, flagella, that type of thing that we covered. You see this illustration a lot. It gives you an idea of the condensation. It doesn't really show the plasmids. We have other illustrations that will do that. Here's the ribosomal unit that you can see there. Again, the conversions and the name for Svedberg units. The same with the cytoskeleton, storage granules, different things are there. The metachromatic ones are interesting because with methylene blue, which you use in the lab a lot, they do have to have different colors, and it's a way of identifying a species called Corynebacteria, which you may or may not work with. Let's see, here's the endospore thing. Germinate to become, so they revert. They either go from endospore stage to vegetative stage. No in between. They're either vegetating and reproducing or they're endospore forming. And then they talk about the process, 8 hours, etc. We can do this in the lab, typically when nutrients are available. when the conditions that were adverse are restored to normal, then they begin to flourish. We will see all of this played out in something in the next major chapter that we do called the bacterial growth curve, which is really a foundation for understanding and for treating and addressing the problems. Again, it says not a means of reproduction, was that little nugget under there, which brings us here to the eukaryotic cell. And that's really, it's very much a review because you spent the last term, most of you, looking at eukaryotic cells in AP1. You see, I started to look through this, and that endosymbiosis is probably the critical piece of all of this, I would think, for new material. That's there. Okay, 2 or 3 billion common. We'll look at this last common ancestor. Single-celled and independent over time form colonies. That's multicellular. They're like us, more specialized, etc. We'll get to that. The circular chromosome is certainly what they're going to be talking about. So we're going to get to endosymbiotic theory and all these kinds of things. To me, the two major things that we see associated with evolution that are really, to me, more scientific than the Darwinian view. Darwin looked at a chain of islands that were initially attached and became separated due to conditions of the sea, and looked at different, and effectively looked at different birds and different beak structures, and basically related his, and he was right that basically descent with modification what it was. Well, though you can poke holes in that, and a lot of science, that's what scientists like to do. But what we do is we see that, you'll recall this is the 9+2 that we find in cilia and flagella, let's say in our cells. We see that in the most primitive eukaryotic cells, in protozoans, flagellated protozoans, which cause us a significant degree of problems in any event. But we see this pattern the same way we see it in us. The DNA has, basically, when you analyze the strands molecularly, it has the same kind of patterns and sequences that we see in our DNA. Okay, so those are consistent. It's good old double-stranded DNA, A's, G's, C's, pairs of chromosomes, and 9+2 apparatus that we see. That's a pretty consistent finding, and that alone is enough to support the descent with modification and the advancements of different species. So eukaryotic flagella, so some of the, and to me, highlight the distinction. First of all, bacterial flagella are not made out of microtubules, eukaryotic flagella are. So it's a different protein, it's called tubulin, where they describe this interesting protein called flagellin. And everything's bigger in eukarya, 10 times thicker, more complex, encoded in membrane. They're not hairs protruding, long sheath cylinders, regularly spaced microtubules. The cilia, similar, obviously smaller, but more numerous, found only in single group of protozoa and certain animal cells. There's a real distinction. In flagella, eukaryotic flagella, we only, by and large, we see them in the sperm cell, other than in the more primitive forms of protozoans. Glycocalyx. We have similarities in that in eukaryotic cells, whether they're networks of fibers, slime layers of capsule, and so there's not an extraordinary degree of distinction between them. Cell walls. Fungi, which we look at a bit here. Yeast and fungi, cell walls. Algae, cell walls. They're very different, okay? Other than that, because there's a lot of distinction, but they do have a cell wall. Okay, and you remember when I presented that to you earlier, kind of at the end of the first week that we had, not last week, but the week prior to that. When we look at ones that are in water, or ones that are photosynthetic, or different aspects of them, is where we look for distinctions that are there. Protozoans, which we talk about, helminths to me are the worms, and to me they're more macroscopic. They start microscopically, but as they aggregate together, they become macroscopic or visible. And I don't know enough about it teaching anything. And I say it again and again. If you want to know about worms, talk to the veterinarian. they're into that. So, protozoans, very, very important, as I've said before. So, cell walls of fungi, rigid shape and structure, different chemical composition from bacteria or archaea, okay, polysaccharide fibers, and this is the one that's interesting. This chitin cellulose is certainly associated with plants. Understand fungi and yeast are it's almost, you could call them plants because of a cell wall, but you can't because they don't have any chlorophyll. And you can't call them animal cells with regard to the cell wall. So they occupy a very interesting niche, so to speak. So they have cellulose similar to plants, and chitin is the material that insect shells are made out of. And again, on certain kinds of mushrooms, when you have that thick stalk that's inedible, it's made out of chitin. Good for stock, lousy for eats. Okay. You don't make your own vegetable or chicken stock, I take it. See, the magic of the Costco chicken. The $4.99 rotisserie chicken, you get like 2 meals out, and then you can use the carcass to make stock. And it's delicious. A little thyme, a little bay leaf, a little mirepoix, happy days. Okay, moving on. So, again, and these mixed glycans. So, really, the critical part with this are two. One, the chitinous area underlying the cell membrane, giving it shape and rigidity. And the other thing is the ergosterols. Those are the particular I don't know if they mentioned it there or not, but the particular sterols are part of it. So here it is. Typical bilayer phospholipids in which protein molecules are embedded, like us, contain sterols of various kind, rigidity and shape to the membrane. Very important when they do, even for cells that don't have a cell wall. Again, I wrote it down before, but the sterol that you have to be cognizant of in fungi and yeast is this. And because it's unique to these structures, that's the big target for antifungal medication, ergosterol. That's the good news. It's relatively unique. The bad news is it's still a sterol, and those medicines are very, very harsh. That's the problem with when we try to address eukaryotic infections, like fungal infections, yeast infections, protozoan infections. They're very similar cell structure to ours, so they typically have a lot of side effects. Sometimes the side effects are quite severe and deadly. Okay. Nucleus you're aware of. Okay. Membrane-bound organelles. This is all the same stuff. The pores allowing the transiting of a variety of materials, including RNA materials to enter, RNA to exit, etc. Migrating through the pores. Our old friend, the nucleolus. Remember the test question. Ribosomal RNA. Yes, synthesis. Everybody says the same thing. Chromatin. Again, these are not those compacted types of chromosomes that we saw there. Okay, these are not supercoiled per se. These have to do with the association with histones, so they're chromatin, and again, when they're visible, they're chromosomes. So we've seen that movie before. The endoplasmic reticulum, and again, membrane tunnels. Yours is all familiar to you, so we're buzzing through it. The RER, and the distinction here, let's say with bacteria, when they have ribosomes, they're all making proteins generically, okay, that are there. Some are going to inevitably going to exit the cell, but we don't have that same degree of sophistication with the transiting of things for membrane-bound organelles. So we don't necessarily have the elaborate nature. That's part of it. So that simplicity, again, I think that is able to dedicate most of their aspects to reproduction. And I think most of the stuff that exits are waste products that end up we call toxins that are part of it. So what makes the distinction here? Because there's no membrane, the ribosomes can't bind to it for specific purposes to make it rough endoplasmic reticulum. So there's neither rough nor smooth that's there. And we know about the jobs that they do, smooth more about lipids and non-protein macromolecules, etc. And again, that again, we see this in bacteria where you might see something similar like this, but it just does not occur. It occurs within the cytoplasm. So again, they sometimes will refer to these as polysomes or polyribosomes. You always see the same illustration for it. Okay. And so the Golgi apparatus, packaging and shipping, you remember that. Different kinds of vesicles, not a big deal for us with regard to this. It's a kind of a neat way to express it with this nature's assembly line, nucleus, endoplasmic reticulum, Golgi, da 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 da, and then basically transporting it out encased in membranes to protect it, and typically expelling it. It could stay in those membranes, or it could be associated with the plasma membrane itself. Again, these cells, lysosomes, which are stay within the cell, intracellular digestion, and for defense. You should remember that. We see in microorganisms a few more vacuoles. Okay. Uh, we see them more associated with phagocytosis, but they also are, again, a useful way of identification for some of these that are there. And I don't want to really go over this, but just review it yourselves in whatever method you wish to. Things that are ingested in eukaryotic cells, okay, by endocytosis, which they do, fuse with lysosomes. They form that phagolysosome. If it's a nutrient, they digest it and utilize it. If it's a bad guy or something that's harmful, hopefully they destroy it. They have to spit out the residual material that's there. And effectively so they have a similar kind of defensive structure the way any of our cells would have that. Mitochondria. There are two energy-related organelles, and that's where this endosymbiosis nugget that's so important comes from. That's, again, sort of the evolutionary nugget as to how the common ancestor effectively became where effectively we got the two lines, the prokaryotic cells and the eukaryotic cells, how membrane organelles kind of got started. They are both energy producers. They both have the capability to make more of them or to go away, okay, and make more when necessary. They will both have their own DNA and their own RNA. They're like little organisms sitting inside of one of our cells. They are, okay, mitochondria and chloroplasts. And the likelihood is they come from sort of generic prokaryotes, let's call them generic bacteria, versus generic pigmented bacteria, which typically are pigmented with some form of chlorophyll. And so that's the lineage, so to speak, as you'll see. So the mitochondria, again, double layer of membrane, which is another one that kind of points to endosymbiosis that's there. The outer membrane, the inner membrane folded, the cristae, because that gives it more surface area for all the aspects it's going to do of aerobic respiration, all the chemical energy. They divide independently of the cell, make more or don't make more, just let them degrade until we need more. Circular DNA, the same kind of DNA we see in bacteria. Bacteria size 70S ribosomes. The ribosome in the eukaryotic cell is 80S. This is 70S. So those are the two critical features. 70S ribosomes and the circle of DNA. Those are prokaryotic structures, no question. And that's why when we address agents that we use agents to impede bacteria that target protein synthesis, and that's a vast category. I mean, the Z-Pak alone being perhaps as prescribed as any antibiotic other than the penicillins and cephalosporins, they target that 70S ribosome. We have them in our cells. That's why we have side effects. So you've seen those kinds of things. They're showing you the 70S. Somewhere there's some DNA that's in there. It's generically speaking a circle, certainly not in pairs. The chloroplast, algae and plant cells, different kind of energy transformation, photosynthesis. Oxygen gas is the byproduct, resemble mitochondria but are larger, and they have pigmented materials that are there in the form of chlorophyll. The ribosomes, scattered, as we know, free, and the ones that become rough endoplasmic reticulum when they become bound. Again, the polysome formation, large and small subunits of ribonucleoprotein. So they're RNA and protein structures. Their combined density measures to 80S with a combination of 60 and 40 when they condense together the small and large subunit. Yeah, well, and then the cytoskeleton, the skinniest, the actin, the strongest, the insoluble intermediate filaments, the microtubule, the largest, long, hollow structures that are there. You've seen this type of stuff, and you can look through these. There's not a lot to say about it. This is the endosymbiosis. So they presented here interestingly. They call this the last common ancestor. Again, I don't know anybody was around. Even the 2,000-year-old man wasn't around then. No 2,000-year-old man fans? Go back and YouTube 2,000-year-old man with Mel Brooks and Carl Reiner, the late Carl Reiner. They did it as a gag at a party one night, and it went from there. Is likely a large organism with loose boundaries, has RNA as its genetic material. That's the theory. When they say likely, nobody knows. Individual cells form genetic material, becomes DNA. You have viruses that change, like HIV, RNA to DNA. You have now, as we can see what's happening, you either have some that are DNA, some that are some that are in the archaea family, some that are in the bacteria family, and then these bacteria are engulfed by other descendants, okay, and they become mitochondria. And the theory is that it's more likely archaea was our predecessor because the DNA sequences are more similar to ours. That's where that aspect of the theory comes from. And again, it's not a big deal for us, okay, but I'm trying to give you the science behind it to some extent. And then they become, whether they have pigmentation or not, either mitochondria or chloroplasts. That's endosymbiotic theory. So there's going to be a question or two about that. The fact that I'm not going to go crazy, was it bacteria or archaea where the original way it formed in, but you should know about the 70S ribosomes and the circular strands of DNA. We'll move on to some of these more specific. Here's the fungal nuggets. The difference between yeast and mold versus when we look at a mushroom, which are all part of the same wider group, what we call fungi and yeasts. We can see mushrooms. Okay, the microscopic forms are yeast and mold. What's interesting is that when they can flip-flop between those, down the road when we get to respiratory diseases, you're going to hear about something called dimorphic fungi. So it means they can flip-flop between yeast and mold. Those are typically very pathogenic. There's a lot of very severe respiratory diseases that come from that, okay? Asexual reproduction. Basically, they bud, and budding is something that you might see in laboratory if you start to look at yeast. Where you see these little yeasts are fairly circular looking. Sometimes they have like a little mini appendage. It's one of the ways they bud. When they form mold, they have hyphae, and that's one of the ways we identify them. And there's a lot of different ways that they replicate. Okay, and sometimes we'll see chains of yeast cells. So the dimorphic, that's because what'll happen, you'll inhale them as yeast, and then they'll form these hyphal strands in your lung, and that's where they cause the problem. So this is when they start to look at different kinds. So you can see this is called rhizopus, which sometimes I'll find on strawberries when they get moldy, and then penicillium, which is bread mold. That's there. Okay, and so you can look. This is the budding the budding process that goes on. These little reproductive structures that form. 300 species can cause human disease, commonly acquired by environmental pathogens, community acquired rather, by environmental pathogens. So it's not unusual to see these. They can be hospital associated, and they're very much opportunistic. You're going to find most famous of these historically. The one that we probably see the most today is called Valley Fever. It used to be called San Joaquin Valley Fever. And if you live in the Southwest, you know all about it. You live in Arizona, you live in New Mexico, you know all about it because you can get it. Your pets can get it. Okay, a lot of people get it because it's in the air. There's the nature of that dry desert air gets a lot of particles that are in it. They're there. It's not the only one, but it was one of these diseases that became the first AIDS defining diagnosis called PCP pneumonia. That's what the original definition back in 1981, when AIDS was defined by this people with immunological diseases, they found this unusual type of pneumonia developing. It was an opportunistic infection. So AIDS patients, fungal cell wall gives of substances trigger allergies, obviously poison mushrooms, different thing. Aspergillosis is not very good. Okay, aspergillus uh, is something that you can also come in contact with, and you can inhale this. And a lot of times, we saw it in one of my stepsons. It was at Slippery Rock, and he was in an apartment, and they like to put carpet on top of like concrete, and it got wet. And when he moved into the apartment, they didn't change the carpet, and the smell was coming there. And we cultured it. Okay. They can affect a variety of crops that are there. That's very common, as well as the animals. Okay. So you could say fungi cause a lot of problems from a commercial sense as well. They're decomposers or saprobes. Okay. They play an integral role with a variety of plants, and they play a big role with regard to fermentation for alcohol, vitamins, antibiotics, and I'm a fan of mushrooms, but that's from a standpoint of, and they are some fungi and related species are some of the most sought after in the world that are there. Effectively, a truffle is a variant of that, which is probably of all the sort of luxury foods that's high up on the list. We'll stop there. Thank you very much. Or as I'm fond of saying, truffles have riches. Oh, nobody cares. Nobody thinks that's funny.